Center for the Ethiopian Educational Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson on the classification of bacteria. In our last lesson, we began our unit on microorganisms. We discussed the microorganism bacteria. We looked at the structure and shape of a bacterial cell. Today, we will continue learning about bacteria. We will use the knowledge we gained in our last lesson about bacteria shape and apply it to classification methods. We will also learn about the Gram stain, which is another method used to classify bacteria. To begin this lesson, let us do a review activity. Students, can you name the 3 bacteria shapes and then describe each shape?
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. I am sure this was a simple activity for you. The three bacteria shapes are cocci, bacilli, and spirochetes. Cocci are round bacteria. Bacilli are rod-shaped bacteria. Spirochetes are spiral or corkscrew-shaped bacteria. Did you remember all three shapes? Bacteria cells are single, but sometimes two or more can be connected. Since there are so many different kinds of bacteria, they need to be classified so biologists can recognize and learn more about them. Before we discuss the ways in which bacteria are classified, let us talk about why classification is important. Students, what is classification? Why do you think it would be beneficial to classify bacteria? Please discuss these questions with a partner.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, everyone. I am sure you had some interesting ideas. Classification is a method that scientists use to group and categorize organisms by biological features. The classification of bacteria helps to identify organisms according to groups. These groups contain organisms that share common characteristics compared to organisms in different groups. A simple way to classify bacteria is by shape. When a biologist examines bacteria under a microscope, they can make an immediate observation about the bacterium based on what it looks like. Another way bacteria can be classified is according to the Gram stain. This is a test that classifies the bacteria into one of two categories based on the reaction the bacteria have to the stain. Bacteria can be stained gram-positive or gram-negative. If bacteria are stained purple by Gram's stain, they are considered gram-positive. If bacteria are stained pink by Gram's stain, they are considered gram-negative. Since Gram's stain produces different results with different types of bacteria, it is called a differential stain. A differential stain is a test that uses staining to classify organisms or organic material. You may be wondering why bacteria stain differently. This is due to the structure of the cell wall. The bacteria that are gram-negative have less peptidoglycan in their cell walls. Peptidoglycan is a complex molecule made from sugars and amino acids. Peptidoglycan is the part of the wall that absorbs the stain. Gram-negative bacteria also have a membrane outside the peptidoglycan that gram-positive bacteria do not have. This membrane secretes endotoxins, which are toxins that are a structural component of these bacteria. Endotoxins are very resistant to antibiotics, making diseases caused by gram-negative bacteria harder to treat. Overall, gram-negative bacteria cause more serious diseases than gram-positive bacteria. However, there are always exceptions. For example, bacteria that cause tuberculosis is gram-positive. Students, let us do an activity to review what we have just learned about gram staining. In the following statements, please determine if each statement is true or false.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. How did you do? Let us go over the answers together. The statement that bacteria stain differently due to the structure of their nucleus is false. The cell wall determines how bacteria stain. The statement that gram-positive bacteria stain pink and gram-negative bacteria stain purple is false as well. Gram-positive bacteria stain purple, and gram-negative bacteria stain pink. The statement that endotoxin is a structural component of gram-negative bacteria is true. The statement that gram-positive bacteria have more peptidoglycan in their cell wall is true. The statement that a differential stain is a stain test to classify organisms is also true. Students, did you answer them all correctly? If so, great work. This brings us to the end of the lesson. Today, we discussed the classification of bacteria. We explained why it is important for scientists to classify bacteria. We also learned that bacteria are classified by their shape or by Gram's stain. We learned that bacteria classified in Gram's stain can be either gram-positive or gram-negative. Until next time, thank you, teacher. Thank you, students.